In this video, we'll take you through some of the resources available inside the VXR labs for enzymes and enzyme functionality. They have some things in common, and we'll point out some of the equipment that you have in there. Let's first begin with how you adjust your settings when you first arrive in the lab, because you may, for example, find yourself at a different height than you would expect when you're working at a laboratory table. So once you're inside the lab, you can twist your left wrist and it will show you some different choices, notably one of them is menu. When you do that, you can flick through the different menu options, but you have this little settings option, and that brings up volume, microphone, tracking mode, etc., etc. So you can look through those choices, but in particular, if you choose tracking mode, you'll see the option for whether you as an avatar in the room are seated or standing and what your height is. And why this is of importance is because you are operating at a virtual laboratory table. So if you are particularly tall, you may find it challenging to reach down and grab the bottles or pipettes, or vice versa. If you are a little short in the room, then you might want to adjust your height so that you can reach to the top of the table. So that is something that you want to adjust as soon as you get into the lab. Now we're going to take you on a tour around the enzyme functionality lab, but note that a number of the resources and the way in which you interact with them in this lab is very similar to the enzymes lab, so you can note these skills and use them in both virtual environments. So once you're inside the virtual lab, you can move yourself around the lab and towards the screen that will contain the instructions for the lab that you have selected. Note that if you're not used to being in the virtual environment, moving around the lab quickly can trigger a little bit of motion sickness. So go slowly to start with until you get used to being in there, and take your time. Now, if you are in the enzyme functionality VXR lab, you will see several different choices at the top of your screen. So there's options and instructions to carry out experiments in regards to the effect of temperature, concentration, and pH on enzyme functionality. Now be sure to refer to any directions given to you by your instructor as to how to interact with these, whether you're expected to do just one of them or all three different investigations, or whether you're expected to adjust your approach to them in any way. Now let's take the effect of temperature as an example and see what materials we have available. So if you take a look on the lab bench to your right, you'll see a number of bottles and a number of pieces of equipment for you to use. Catalase is the enzyme you'll be using in this case, and you'll need to put it into the test tubes. So that will require use of a pipette, so you'll need to navigate around the lab table and pick up the pipette. Note that you shouldn't move through the lab table, because even though you could in the virtual environment, you will knock over all of the bottles and then you lose some of your equipment. Now, to pipette some of the catalase into the test tube, it's important to note that you're not taking the lid off the bottle. Instead, what you're going to do is put your pipette by the top of the bottle and use your trigger finger in order to take up the liquid. And you'll hear a little sound to indicate that the catalase has been taken up into the pipette. So note that, because otherwise it might be a bit tricky if you spend a long time trying to take the lid off the bottle. So in this example, since we're doing the effect of temperature, we have now added 1 drop of catalase to 3 separate test tubes. Note that 1 plastic pipette in a real laboratory would contain 1 milliliter of catalase. So if you're being asked to plan an experiment or note values, 1 drop of catalase would be the entire plastic pipette, and that is 1 milliliter of catalase. Now note that in the next step, you're asked to place 1 test tube on the thermal modulator at a given temperature. Now, the thermal modulator is a piece of equipment in the virtual lab that is equivalent to, say, a water bath or an incubator in a real lab. Either way, you can adjust it to a given temperature and allow the reaction to take place at that set temperature. So now we're going to navigate over, pick up 1 of our test tubes, and go and place it on top of the thermal modulator. So once we've placed it there, we can turn it on using the red button, and with the dial on the left, we can adjust the temperature as needed. Once the temperature has been adjusted, we can check that our test tube is at that temperature using the thermometers next to it. So if we pick up one of the thermometers to the left and we hold it over the top of the test tube, you will find, if held correctly, that it shows the temperature that the test tube is at. And you'll also hear a little sound to indicate that it is measuring the temperature of your test tube. Navigating over to look back at the instructions, we note that it is also asking us to have a test tube at 100 degrees C. In this particular lab, we can go over and we can place another test tube on the other thermal modulator. So what's important to note is that you're following the directions on the screen in the virtual lab or any specific directions given to you by your instructor. So navigating again back over to the directions for the lab, we note that those directions then also inform us of how to add the substrate to the enzyme. And in this case, since the enzyme is catalase, the substrate is hydrogen peroxide. Therefore, we need to put all of our test tubes on the one bench where we're going to be operating, just like you would do in a real laboratory. And then you're going to go and obtain your substrate, hydrogen peroxide, because we're going to want to add this to our test tubes and observe and measure the reaction. Two things to be reminded of here. First of all, in order to put the substrate into the test tubes, you're also going to need to use a pipette. So remember once again that you're not trying to unscrew the cap of the hydrogen peroxide, but that you're going to use a pipette and use your trigger finger once the pipette is held over the bottle cap of the hydrogen peroxide. That's how you're going to take out the liquid. Hover the pipette over the top of the bottle, use your trigger finger, and then you will hear the noise that the solution has been taken into the pipette. The second thing to note, for the reaction to take place in this virtual lab, we need to stir the contents of the test tube. So at the point at which these things are put into the test tube, the reaction hasn't happened until you use your stirring rod. Now this is stated in the instructions. It does say add hydrogen peroxide and stir, but I do think the stirring portion is a part that can be overlooked by students, and it's really important for you to know when the reaction is starting to take place, you're also going to observe bubbles of oxygen being produced. This is slightly different from outside of the virtual lab, whereby as soon as the substrate and the enzyme come into contact with one another, they would react. So that is slightly different, but when you go ahead and stir, you will notice that the bubbles of oxygen start being produced, and then you can use your ruler to measure the height of those bubbles in the test tube. And the height of those bubbles is what you're going to record as your dependent variable. Remember that you're going to record that height in metric units, so that's centimeters, not inches. So now you've familiarized yourself with the virtual lab, it's also important to know some of the other functions that are available. For example, if you do knock over a number of bottles and they smash on the floor, or you do run out of some pieces of equipment, what should you do? So there is a screen that is available to the host, and that would be you if you're in there by yourself. And this allows you to gather all the objects on the floor to also respawn or recreate objects that were broken. Now there is one more piece of equipment in the lab. It's called the fabricator of the virtual lab. And this is a piece of equipment that allows you to generate more of any piece of equipment that you've run out of. For example, if you'd run out of enzyme or substrate, normally your technician would get more for you, but here in this virtual environment we have the fabricator. So you get a chance to select which piece of equipment it is that you need, that you ran out of, and then you hit the red button in the middle and it is going to fabricate to create that piece of equipment for your use. So in this case, a test tube, each one of them does different pieces of equipment. This one, you have your catalase, you have your water bottles, your hydrogen peroxide, and you can select whatever it is that you need. And there you go. It is fabricated for your use. So no need to worry about running out of any pieces of equipment.